does have authority. They give it to you because of export. That doesn't mean to say it's bad to use it, or at least make it known what it means, because it really raises the bar.
Right, absolutely, yes. Now that's very important. And I do want to go back to that mTOR pathway, because I know you had published an article about like the stress and fear response in autism that we often see with kids having higher stress responses. And is that related to the mTOR pathway, or what's the connection there?
So the paper was published by pulling basically data published by other colleagues. So we know that children are more stressed out. They're more anxious. We also know they cannot handle stress, very well. We also know that one of the major risk factors for a child developing autism was severe stress of the mother during gestation. Now, if you link all of that, and that's where our contribution came in, with the fact that we published separately that CRH, the first hormone released under stress, can trigger the mast cells of the microglia. Putting the two together, we basically said we know that stress could contribute to whatever is happening in the brain via these means. But we did not actually look at mTOR specifically in this or any other study. However, what we did, and we published this in two different ways. One was published about 6 years ago in the Proceedings of the National Academy of Sciences with colleagues basically, and we looked at brains from children that had died and that were on the spectrum versus children that were otherwise normotypic and they died pretty much the same way, or drowning or car accident or whatever. Looking, of course, at the quality of the RNA, so we wouldn't, botch the results. And it was apparent that inflammatory markers were actually much higher in amount in the part of the brain that regulates behavior, which is amygdala.
Okay.
Then, just about a year and a half ago, with colleagues from the University of Mississippi, we looked at the whole genome of the brain of children, again, with and without autism that had died. And of course, when you look at the genome, that indicates that the genes were activated, but that doesn't mean necessarily that they will make the inflammatory proteins.
Right.
But there were two clusters that stood out. One cluster of genes was the genes that make connections between nerves, and that was way low. The other cluster was inflammatory genes, was way up. And it was only, again, in amygdala. So that is telling us basically in different ways that there is inflammation, and the inflammation either contributes or happens to be a parallel finding with the fact that the neurons just don't communicate properly anymore.
Right.
It's like the spark plugs got rusty.
Was that the inflammatory mediators like tumor necrosis factor, interferons, just various?
Perfect question. So at least we looked, and in the brain, it was interleukin-1 beta, interleukin-6, interleukin-18, and TNF at various levels. Okay. Most importantly, we were the first ones to publish this. We showed that both mast cells and microglia in the brain samples were positive for an enzyme called MMP9, matrix metalloproteinase 9, and that disrupts basically the connectivity, just kind of cleaves the scaffolding. So then we went back to the laboratory and we stimulated, as I said we did earlier, either mast cells or microglia with the triggers that we know or suspect they might stimulate it, and we measured MMP9, and MMP9 came out. And then we pre-treated the cells with luteolin, and we blocked release of MMP9 as well. And now we're actually in the process, we have two even better flavonoids that might, we might be able to put in some supplements. It's just that difficult to purify them. And if there's no demand, the price is much higher, obviously.
Yep. And so those flavonoids, like the new ones and the ones you're currently using, those are decreasing those inflammatory markers.
All of them.
All of them. So MMP9 too?
Yes, from both microglia and mast cells. So you get basically two birds with one stone, if I can call it that. And as you know, because you alluded to this earlier, luteolin now has come up to have additional benefits. Of course, it's antioxidant, etc. Yeah. There are 3 papers just recently that it might have tremendous anti-cancer activity.
Wow. So who knows what the actual use will be? It could be, everything, all health conditions. If there's inflammation going on or present.
Yeah, it's just that because they're natural molecules, so they're not patents, companies are not interested in them. That's the problem. Otherwise, 20 years ago when I started studying with flavonoids, I remember I had visited a company in Europe because they make this, back then they were not as popular power bars, they might honey and nuts, whatever they might have. And I said, well, if we fortify, milk or cereals with vitamin D, why not fortify them with luteolin? It's anti-inflammatory, it's antioxidant, why not? And even though vitamin D doesn't have a patent either, well, that's a vitamin, so it's a little easier than to go that route. I still think it would be tremendous, to have 45 cell foods with it, and you don't have to kind of worry about it.
Well, especially because if we think of the toxins that we're exposed to now, which didn't exist years ago, now we're overwhelming our bodies with these toxins, and then we're increasing oxidative stress, and then that's triggering the inflammation. It's just this cycle, this nasty cycle, right? So it really needs to be broken up where we're decreasing that oxidative stress, and you're saying that this is one of those ways you can do that.
Yes.
These flavonoids. Yeah, no,
but I have to stress the fact that not all flavonoids are good. So you see terms out there, isoflavonoids, soy flavonoids. God forbid if someone has estrogen-dependent cancer, you don't want a soy flavonoid. That's going to make things worse.
Right. So very personalized or independent.
Yeah. Right. The other thing is I think I kind of said earlier, but I say it again. If you give someone, colleagues will say, well, you don't absorb very much, quercetin, whatever. They'll give you 2 grams a day. So you absorb only 10%. That means 200 milligrams, 1,800 milligrams stays in your gut and now shuts down everything. And now you've got dysbiosis. You start chasing your tail. So too much of something is not necessarily good. I always say, start with the least amount that you can tolerate and then slowly build it up to the point that maybe you get some side effect, whatever that means, quote unquote side effect. But don't go full blown, highest dose possible, because you never know what happens.
Right.
For instance, you might have a young girl just entering, let's say, puberty, I don't know, 10, 11, whatever they are these days, and you give them 2 grams quercetin, it's going to shut down the liver enzymes. The estrogen will go off the roof because it will not be metabolized. So you might have now precocious, puberty all of a sudden. So there are all kind of things that we don't really think about when we just go, I don't mean you and I, go out to buy a supplement.
Yeah. So always, I would say low and slow, right? Start low with the dosing and then increase slowly.
Absolutely.
Yeah. Have you done any studies or looked at studies with oxidative stress and mitochondrial dysfunction?
Not directly. I have colleagues who look at that, but we published something related, uniquely interesting. So mitochondria, as some of the listeners may know, were actually bacteria that became symbiotic with our cells millions of years ago. That's why their genetic information, the nucleic acids, are different than the nucleic acids of our cells. So the nucleus of our cells has different nucleic acids than the ones that are actually in mitochondria because they came from bacteria. Number 2, usually the mitochondria are around the nucleus if you look at cells. So I had a very bright graduate student doing his doctorate thesis with me, and he basically argued with me that mast cells have a lot of mitochondria and they do a lot of things. And I said, well, the mast cells are very quiet. They don't multiply, they don't run around. So why should they have a lot of mitochondria? Anyhow, it was a bet. So we used electron microscopy as we did, and of course there were lots of mitochondria. And then we said, well, what if the mitochondria actually do more than just making energy? And we asked the question, that's how science works.
Yeah.
What if the mitochondria, if they were misbehaving, call it damage, call it whatever, if they get fragmented and they dump their DNA outside the cell? So we stimulated the mast cells and we look at them on the microscope, electron microscope, and what do you know? The mitochondria that might have been, for instance, this big around the nucleus became fragmented to what the cap is only, and they were all lined up along the surface of the cell. We were absolutely amazed. So fragmented mitochondria came to the surface. We published this numerous times. And then we said, well, what if the mitochondrial spills out its content? And then we took the culture medium, basically, and we measured mitochondrial DNA versus what we call genomic DNA. Mm-hmm. So if the cells were dying, then the genomic DNA will actually be dumped outside as well. And it was only mitochondrial DNA. So then we took mitochondrial DNA and we added it to the other culture cells, microglia, neurons. They stimulated the hell out of them. So what happens is when mitochondria get damaged, two things will happen. Either they will dump their cargo outside, or they will undergo phagocytosis, meaning the cell itself will destroy them internally. And if they release their content outside, the body misconstrues this DNA as a bacterium, basically, because that's what it was. And it mounts an autoinflammatory response. So that's another problem that we actually have with.
It's just it's that cycle. It just seems like it's just a nasty cycle that keeps.
And it gets even more hotter because we then measured free mitochondrial DNA in the blood of children on the spectrum, and it was high. Okay, so usually you see free mitochondrial DNA in boxers, where you hit basically the muscle, and of course some cells will be destroyed and DNA might be released. In this case, there was nothing. So, and we published about 7 papers on this, but for some reason this has not been picked up, furthermore.
No, and this is a huge piece of the puzzle that could just be helping so many people, I feel like. So, we've talked about so many things in relation to autism and how it all kind of comes together in this nasty cycle, I guess you could say. But from your research, would you say autism spectrum disorder is, with that umbrella of symptoms, being caused by environmental toxin exposure and accumulation, and then that trigger in that cycle of inflammation, mitochondrial dysfunction, oxidative stress, and all those factors? Is that kind of what you have seen, or in terms of what autism spectrum disorder?
You're saying that because we have a paper that again asked for some last-minute corrections to other colleagues, and the title is basically "Role of Mast Cells in Gut-Brain Axis," and we talk about toxins. So the answer is basically threefold. Number one, I don't think that that autism is a disease. I think something happens perinatally that causes the brain to reset itself in a kind of a fight or flight mode. Right? It's like there's danger all over the place for whatever reason. So the microglia just sense something that they shouldn't be sensing. That's number one. Number two, I really think it's a stimuli, if you wish, condition. It could be toxins, it could be infections, it could be stress, and it could be some genetic predisposition. Mm-hmm. What I take issue with is colleagues, and they're wonderful colleagues, and they're wonderful supporters. The Simons Foundation in New York gives a lot of money, but they focus primarily on genetics. A family gave $50 million to the University of Pennsylvania, I think, recently to create a center for autism. But both the director and the emphasis is, again, genetics. An issue was published a few weeks ago by Time magazine. It's all about autism. You can find it on the stands. Mm-hmm. And it's very well written, but again, the emphasis is it's all genetic. That's not true. I don't understand where that comes from. Because
would you say it's definitely a genetic susceptibility where you have certain variants that make you more susceptible to those toxin effects?
So there are three. Let's just talk about genetics first. Okay. Yeah. So the 2 studies were published many years ago where they looked at identical twins and non-identical twins in the general population. Even the identical twins, if a child had autism, the other child having autism was only 40%. So and they were identical twins. So if it's absolutely genetic, then it should have been 100%. So
100%, right.
Second is that there are about almost 300 genes that have been found to be more common in individuals with autism. But that doesn't mean to say that they will cause autism. They're more common. Now, does that mean that there's susceptibility because of those genes? There may be. And therefore, we talk about what we call epigenetics. Some of those genes or cluster of genes might be quiet, and now because of toxins or whatever else, they wake up and they create the problems, kind of they do. Yeah. And then there's some chromosomal abnormalities that obviously involve genes, where there might be tuberous sclerosis, Fragile X, Rett syndrome, Pett syndrome, and there, 60% or so of the people that have these genetic problems do actually have symptoms of autism. So that's a little cleaner, quote unquote, to study. The point is, if we actually say, yes, there are genes X, Y, and Z, it becomes ethical. So what do we do with this now, since they've never been shown to definitively cause actually a problem? Now, it wouldn't surprise me is over the years, it is identified that certain gene mutations do lead towards that direction. Right. But so far, none of those genes is really proven to be definitive. Yeah. Now, one question is, how about genes that regulate neurotrophic factors? And there's nerve growth factor, and there's NGF, and there's BDNF, brain-derived neurotrophic factors. Those have been shown to be lower in a condition called Rett syndrome, which, as I said, et cetera. What is amazing is the flavonoids we were discussing have been shown basically to stimulate production of these factors in the brain. Wow. So there might be additional benefits of these molecules that have not been studied as of yet. And if we have time, towards the end, I can tell you what we're doing now. I'll just mention it, but we're creating, as others have, what is called human organoid on a chip. So basically, we're using a system, in a closed system, where we look at neurons kind of in the middle of a well, and we look at their ability to fire singly or fire together. Okay. In which case we call it synchronized firing, or float off, in which case they're dead. And we measure that through impedance. So this is an instrument produced by a company called Axion through some patents by Johns Hopkins University. But then there are different plates made by another company that works with Axion called Netri from France, where we can put satellite cells in other wells. So what we're trying to do now is to mimic as much as possible what may be happening, let's say, in the brain. So we might take triggers and see what they do to the neurons directly. Then we might take a trigger that is unique for the mast cells, and then see if that releases something that affects the neurons that might not have known before. But now we can take the culture fluid and measure it, and we can take the cells and do RNA sequencing to find out what may have happened there to direct us to measure the right thing. So other colleagues are doing such organoids, especially for Alzheimer's and Parkinson's disease. But I think that's the only way to go, because no cell lives in isolation. They talk to each other and they affect each other. So hopefully there, because we can take the neurons and the mast cells and the microglia and grow them from pluripotent stem cells from the actual patients. So let's say if a patient has some genes that make them susceptible, then in this organoid system, we will see more damage than if we were to use exactly the same collection of triggers in an otherwise normal individual. Wow. That's why this is the only way to prove that there is actually, whether it's a bit genetic or whatever. And of course, using the system, we can test for ingredients in vaccines as well. If a vaccine has X, Y, and Z, we can test each individual component and see if in fact it does damage before it's put in a vaccine, except that no one is funding these things.
Well, yeah, exactly. So you could really use genetics. If you have a certain genetic variant based on what you're saying that is known to make you more susceptible, that, then you could avoid certain toxins because you're more susceptible to having them have those cascade effects from that happening. So if we use personalized medicine, we should see a dramatic decrease in all health conditions because you're factoring that in. You're taking that accountability out.
I believe that as we do with newborns and we test for X, Y, and Z, especially metabolic diseases, we should be taking some Wachal cells and just do the liver enzymes so that we know as soon as the baby is born, can they metabolize toxins or not?
Right.
and then act accordingly. Not wait for 3 years, and by that time we have things happening and we're trying to undo something.
Right.
And it's so easy to do. Yeah, it's so easy to do.
Yeah.
So, anyhow.
And it's cost saving, really, because if you think of all the damage being done and the consequences from that, when you could do something up front, really, yeah. And
it's not invasive. You don't even draw blood.
Right.
Something like
that. Right. That's just amazing. And I did want to talk to you about luteolin, your product, or Neuroprotect. I think you had an intranasal formulation you were working on as well, right? And that was to try to get directly to the brain to have more of an effect. Has there been any progress?
So before I get to that, we spoke about low phenol, and many families, especially with young children, cannot have them swallow, or the children don't want to swallow. They're very finicky. So we created about 2 years ago. Aldo created a Neuroprotect low phenol liquid.
Right.
And that has two advantages. One is you can start with even one dropper, or one drop within a dropper. One dropper is 1 milliliter. So you can start with tiny amounts if someone is sensitive and build it up. The second is now you can mix it up with a little fruit or a little juice or a little, applesauce or something to mask the color, which is yellow, of course, and many children are very sensitive. The third, which is probably the best, is that if you put it under the tongue, literally becomes sublingual, and you avoid distraction through the passage through the stomach. And many families do that now. So that was the next step that we could do. Now, the point with the intranasal, even though we came close to it, is that FDA considers it a drug. So it will be excruciatingly expensive to try to create a new something out of that.
Unless you had a pharmaceutical company pick you up and fund it.
Correct. And the second problem is that these flavonoids don't dissolve in water. Now, the liquid form is in only pomace oil, but you need a lot of pomace oil to dissolve it. So, and they're yellow. So if you were to put it in the nose, it's going to turn yellow as well. Now, luckily, there is this analog that I said, methoxyluteolin, that doesn't have color, which we managed, because it doesn't have color, to put in a skin lotion called Gentle Derm. So Gentle Derm has a number of publications that is excellent for eczema.
Okay.
And for obvious reasons, because it's anti-allergic, anti-inflammatory. Beauty of it is, especially for children that may have also eczema and, the autism spectrum, that they can benefit from that. But many families basically tell me they rub it on the forehead, the lotion. And that's really amazing for two reasons that I can think of. One is that anytime, if the child allows you, of course, to rub their forehead, I've had migraineurs, and if they rub their forehead, they calm down, actually. So if a child is very hyperactive, especially at night, that might be helpful. But it gets absorbed through the skin directly into the temporal vessels. So it's as close to giving it up the nose, if you wish. I'm not skirting the issue. We're still trying to make it intranasal.
Hey, no, that's
that's another possibility. So I would actually recommend someone using the dropper and just put as many drops as they can. And now it exists both with a little lemon, essential lemon taste, just tiny bit. Without it, yeah. Some individuals don't like the lemon taste if they're allergic. It doesn't cause allergies, but they think that it might cause allergies. I mean, citrus fruit could actually cause allergies, but not the essential oil, especially in that amount. Be it as it may, you can have both choices and use basically the Gentle Derm, and you can use it anywhere except eyes and an open wound. It's very hydrating, etc. So,
wow. No, I haven't even thought about that. That's a good point about the using it on the head. So thank you for that. And then if someone does know their genetic variants and they're a slow metabolism, they're like slow COMT, slow MAO, then they would want to get the low phenol product because they're more likely to respond or react.
Unless they're phenol intolerant, if you have actually P450 metabolic problems, you can easily go to 1,000 milligrams a day. Because as it turns out, even with the olive pomace oil liposomal form, you don't absorb more than 50%. You're going to be at 500 milligrams at best. That's not going to affect your enzymes. So you're fine. What I do stress for all our listeners is don't forget that curcumin, pycnogenol, resveratrol, all of those are phenolic. So if you want to find out that you're below 2,000 milligrams, add them all up, basically. So don't just say, oh, this is below 500, whatever, I'm okay. Because if you add them all up, they will actually slow down your enzymes, both in the liver and the gut. So you've got to be careful.
No, thanks for pointing that out. No, I feel like I could talk to you all day.
Thank you. Well, same.
I know we're running probably out of time here, but you're just a wealth of knowledge. I mean, I've really enjoyed learning from you, and I'm sure all the listeners will as well. So thank you. Just thank you for your...
Look, I'd like to leave us all with saying, number one, we should make absolutely sure there's no comorbidity that is identifiable and potentially addressable, say treatable. Because I was asked to see many, many years ago tweens, and there were straitjackets. They were about 2 years old, 3 years old. I said, why are there straitjackets? They had eczema all over their body, and it was scratching their head. But even a normotypic person with eczema, they're going to go crazy behavior-wise.
Absolutely.
So that's number one. Number two, make sure you identify any sources of pain. Pain is a major stressor, and many children are way constipated. And of course, if they're not verbal, they cannot tell you that.
Absolutely.
So you got to make sure. I mean, I had families go and do a flat X-ray of the abdomen, and they were basically absolutely loaded with stool. And so I tell them, if a child doesn't move their bowels for 2 days, you've got to do something. 3 days is dangerous. 4 days is literally unethical, especially for young children, because they cannot tell you they're hurting.
No.
The last part is that many children, and of course, you can't give absolute numbers, but most of the children that I had if I could call it pleasure or honor to help over the years, they don't do well with just one supplement. And you know that, of course. And many families, try various ones. The combination that I found basically the best would be Neuroprotect, slowly increasing the dose, whichever formulation that is. Number two, folinic acid, not folic acid.
Right.
Because 50% of the children have antibodies against the receptors that allow the folate to get inside. And there's a test called FLAT test that you can do. And a company that can send you a kit is called Iliad in New York, and they can do that. And about 40% have mutations on the enzyme that makes the active form, which is the NTHFR. So if you got both, you're doomed. Folinic acid or calcium folinate bypasses the receptors, gets inside the cell through a pump, and doesn't need the enzyme. And there was a post basically on TikTok about 3 months ago by a mother who said, my child started speaking with this unique drug, leucovorin.
Yes, lots of stories about that. It's wonderful.
Leucovorin is calcium folinate.
Yes.
Except that it's prescription. That's the only difference. So calcium folinate up to 1 milligram is allowed as an over-the-counter. More than 1 milligram becomes prescription. And you can buy it either as calcium folinate, generic, or leucovorin, the trade name. And they have some inactive ingredients, so you've got to watch out. And unfortunately, the only company that sells calcium folinate in a solution is actually a company called California Gold, and they're forever, I don't understand why, back ordered. So you might say, why not give the active form methylfolate directly? And there are many companies that sell methylfolate.
Right.
The tricky part is methylfolate, when it's inside the cells, of course, works fine. If we give it exogenously, it still needs to bind to the receptors to get in. So if you've got antibodies blocking the receptors, even methylfolate's not going to get in.
Right.
So I'm announcing Algon is going to have a unique product by the end of September that will contain both calcium folinate and methylfolate in just purified water and glycerin, nothing else, no preservatives, no whatever. Wow. So you can get it both ways.
So you can bypass MTHFR. So if you can't convert to the active form to folate, you're bypassing that, but then you're also bypassing the autoantibodies.
Correct.
So making sure that your cells are getting the active folate.
And it's not going to be back ordered. Formula is ordering thousands of bottles.
Oh, I can guarantee that's going to be very popular.
I mean, I just feel bad because everybody, I say, get Folini, guys, I cannot find it. Then I tell them try California Gold and the forever, back ordered. And there was an issue, but I cannot point my finger to it. I'll mention it generically, not about any particular company. But companies that make a lot of supplements, they use their drums, for one supplement, and then they go to another supplement, etc. They're supposed by FDA, if they're registered by FDA for good manufacturing practices, to clear their drums before going to the next one. So, for instance, let's say they make something that has corn, and some people are, sensitive to corn. They don't clear the drums, and now they make, let's say, I don't know, folinic acid. Well, people might have reactions. So I have a lot of patients that have reactions that we cannot figure out, and they might be traceable all the way back to basically companies. It's expensive to be cleaning your drums, So I'm not saying that any particular company is doing it. I don't want to be accused of that, but it happens in the industry.
Right.
So, how can actually someone know about it? Well, it's hard. I mean, if you're really, careful, you can write to the company and ask them if they can give you a certificate that they actually are allergen free. And that would be a little more reassuring.
Right. Yep, that's a good point too. And along supplement lines, I guess that on like probiotics and prebiotics, because I feel like that's a big area we see too, where there's gut dysbiosis, there's candida, or certain yeast fungal infections, and that kind of all goes together too. Or do you think doing lab testing is essential for these kids to kind of know what supplements they need the most?
Not necessarily. First of all, clearly, I'm a proponent of avoiding histamine-rich foods for anybody, really, especially the children that are sensitive, because they will actually cause problems. It might not be allergic problems. It might be colicky pain, for instance, etc. That's number one. Number two, for those who are sensitive to especially histamine-rich foods, you can use a supplement, diamine oxidase, that breaks down basically histamine, and we use it about 15 minutes before a meal. You just have to keep in mind, because there are many DO supplements, that's an enzyme, and if it goes through the stomach, part of it will be destroyed by the acid of the stomach. So look for any supplement with DAO that says either enteric coated, like enteric coated aspirin, for instance, or acid resistant. Those are more likely to eventually make it into the gut and have their effect. Number three, some probiotics are much better in reducing histamine, and those are the bifidobacteria.
Yeah.
And there's some subspecies like Bifidobacterium brevis, longum, infantis that seem to be better. So I would recommend that anybody who is in that category tries to get those in many different supplements. Look again for any preservatives that might be bothering you. But many of these bifidobacteria are together in probiotics with lactobacilli. The lactobacilli are the most typical ones in probiotics. However, if someone is sensitive or allergic to milk, lactobacilli are from fermented milk. So I avoid the lactobacilli for anybody who's sensitive to milk, because they might then, carry on the sensitivity.
Right. So Bifidobacterium is going to be the best species for histamine intolerance.
Histamine, and it's a good class of bacteria anyhow. So if you've got SIBO and you want to rebalance your gut, because you can use lactobacilli and bifido, except that the bifidobacteria break down histamine, and therefore you have the added advantage of that.
Okay. And then do you feel like that then in itself is going to help heal the gut lining?
Well, I mean, the gut lining is damaged by a number of things, as we discussed. So it might not be just the bacteria, imbalance. So if you've got mast cells firing, if you've got toxins there, it will continue to do damage.
Right.
So that's why I'm a proponent of doing the stool test for the eosinophilic cationic protein calprotectin, because if those are high, any zonulin or gliadin or transglutaminase that are indices of the gut barrier breaking down are high, then we've got to protect the gut somehow. It's not going to be enough. The other, of course, defender of the gut is what we call secretory immunoglobulin A, IgA. And if anybody has secretory problems or gut problems, I measure secretory IgA. Any lab will measure it. Now you might say, so what if it's low? Well, colostrum has IgA. So in that case, we might consider, giving colostrum with some caveats because it might cause diarrhea and other things, but that's the only way to give IgA. So going back to what you so wisely said from the very beginning, the more items we identify, the more likely to find a way to address those rather than just shooting from the hip and giving something and hoping that everything will get better at the same time. It just doesn't work that way.
Right, because otherwise you feel like these kids need just a plethora of supplements because they have all these things going on. It's not just one thing, it's multiple areas that the whole system has been disrupted. And so you're trying to address those. And so that can be super tricky trying to figure out, do they need all these supplements or can we use one or, without doing testing, in my opinion, it's hard to know where to even start,
Sure. And I always say, try one supplement for a week or two. Allow yourself to build it up, see where you are, a month at the latest, and then try another one. I know for many families, because they're desperate, they'll try this, they read that, we'll add this. It just doesn't work. And after all, if you give 30 supplements to a child, that's stressful on its own. So,
absolutely.
It's a way to indicate what one might expect. Obviously, if a child, let's talk about autism for a second, is very functional, it's going to be much more difficult to see the benefit quickly because they're very functional. If the less functional you are, the more likely you will see the benefit from what we're discussing. Yep. Okay? And I forgot to mention after folinic acid, I was going to say, about 40% of the children don't even have vitamin D3, regardless of sunshine, Florida, Greece, California, wherever. So we have to supplement vitamin D3 because it's actually immune regulator. So I usually recommend about 1,000 international units of vitamin D3. So what do we expect? So let's say a child is 3, 4, nonverbal, not necessarily tiptoeing, but flapping when they get excited, etc. Not following commands, etc. So by giving the three things, vitamin D3, folinic acid, and neuroprotect, and increasing slowly, depending on the weight and the age, within a month to 3 months tops, we will see better eye contact. We will see more complicated demands being executed, like bring the shoes from the dining room to the bathroom, that kind of thing. And we start seeing actually single syllables, but used for the right reason. So a child might say no, and they really mean don't do it, kind of thing. By 6 months, I'm talking about 80% of the children I and other colleagues have seen kind of together. You start seeing, a verb thrown here and there, and a lot of echolalia. So start repeating. It might be music, it might be words, it might be the same sentence, over and over. And the verb might be at the end rather than the middle or the beginning, but they start making sense. Okay? At that point, let's say by 6 months, 1 year the latest, okay? Then it becomes a period where children tend to be a little more anxious. And it's a short period of time. I think it's a period where they start realizing they can communicate, but they can't quite do it as of yet, and they get very frustrated. Right. And it usually lasts about a month, and then they kind of progress and they do better. Then there's another, let's say 2 years in, where the parents get upset because they expect the child all of a sudden to be conversant. And I keep on telling them, look, 2 years ago, they couldn't say a word.
Right.
You're not expecting to analyze American politics, The best I would expect is to ask them, can you please tell us what happened at kindergarten or something? And they will actually give you a few sentences rather than being answered. It's like, did you have a good time? Yes. They might say, yes, because I played with such and such. Okay? So I definitely think that in most children, autism is reversible. You just have to give it time. Don't go crazy with, a zillion different things. Stay the course. And if it doesn't work, there's no harm done. But let's not jump, from, 10 different things at the same time. Right. I had an email from a very bright father, the other day that I'll be helping with the child. And he gave me 10 different supplements and publications for each supplement. Should we add all of those? And I said, look, the publications are more scientific publications. They were not really clinical publications. We don't know exactly what they will do. Just give it another year, see where you are, and then let's see what remains. For instance, I have a colleague, a wonderful colleague at Harvard, child psychiatrist, who really thinks about 50% of the children have actually ADD, but you don't pick up the ADD until a child is like 7 years old or so.
Yeah.
And
Yeah.
maybe if that's true, then we can address the ADD separately from everything else, because they might not be all due to the same reasons. Okay.
Absolutely.
So
yeah. No, that I appreciate you sharing that, because being a parent myself and going through this, it's important, I think, for parents to know that this isn't an end all be all. There is
absolutely not.
There is hope. Stay the course, stick with a plan, trust the process, and you just gotta keep trying. But you're gonna help so many people already. You already have. I know it's just gonna be amazing, and I'm so appreciative of that.
You're very kind. Look, I feel for the families, I feel for the children, and I feel for the fact that medicine is so far behind, and also for the fact that, in all honesty, for conditions like this that are multifactorial, we should have like a team of absolute experts to meet every month and just discuss the findings and see where the things go. Even when we go to meetings, everybody presents themselves, but and even if there's like a roundtable discussion, it's mostly questions from the audience that tend to be, of course, personal. What we need to know is, okay, Dr. Theo said, for instance, does Dr. Fry agree? Or Dr. Rossignol agree? And if they don't agree, why? Let's kind of help us.
Have those discussions.
Yeah, and I just picked a couple of names because they're good friends and scientists, but we don't do that. We need what we used to say when I was in Boston, we were watching the Celtics, of course, and it was the dream team. We need a dream team for autism.
Oh my gosh.
As for many other diseases as well. And for better or worse, Secretary Kennedy announced that by September there was going to be such a dream team. Well, we haven't heard anything yet. They promised a lot of money for autism research. We haven't seen that yet either.
So, well, I hope it comes your way if it does become available, because that's, I think, one of the things is that it's going to the right person. It's going to the right people that are doing the right thing.
Oh, thank you.
And you're a good person. And that, to me, that is huge. You've done the research for the right reasons,
That is true. And even I don't know if you know, and maybe we should be saying on the website, but Argonaut supports actually research on autism every year, not with me necessarily, because I don't see children as such, but they give money, for instance, to the University for Research. They give money to the University of Mississippi that funded the study that was published. We looked at the genome of the brain. So these supplements are not going to command money. I mean, the money actually from the supplements to Argonaut literally covers, the salaries, and then the rest goes for research. No one is getting rich out of this.
Right.
And I appreciate your saying, you really need good people to really feel for what they're doing and try to help.
Yep.
Not necessarily just jump on the high wagon, so to speak, and just claim.
Well, and that's why I wanted to do this, and I wanted to interview you, because I know there's so many people out there that they don't have the medical background, they don't have the resources, and I know how much I struggled and my family struggled, and I can't imagine what those families are going through.
Absolutely.
And we just need to do so much better, and to know that there's products like yours and research like yours that can help and make a difference. It needs to get out there. It needs to reach these people to help them, because our current medical system isn't doing that, and it's completely failing.
That is correct. If you go to the site that lists all the clinical trials, clinicaltrials.gov, that's the site, and you look for trials on autism, 98%, if I can say that, of trials is antipsychotic drugs for new indications. In other words, something might be used in schizophrenia, not to be used in autism. There's nothing new.
You don't want to get me started on those, because we've done the antipsychotics, anti-anxiety, stimulants, all those, and because you think that's the right thing to do, but in our case, it made things so much worse. And then the effects that you're having on those neurotransmitters and how you're impacting those, and what you're really doing when they may not even be needed. So that's just, that's a whole other area.
If a child is hyper, and we don't know if it's ADHD or whatever, hyper. I've got to find out why they're hyper. There might be anxiety, and of course, if they have a lot of anxiety, they cannot communicate, and we will consider anti-anxiety natural molecules, like what you're doing with the ones I mentioned. But I always absolutely measure total catecholamines, which is epinephrine, norepinephrine, dopamine, serotonin, in the blood, and the enzymes MAO and CMT. Because if they're slow metabolizers and they're putting out a lot, then they will be hyper because they're putting a lot of norepinephrine. In that case, a drug such as clonidine, which is not antipsychotic, might be helpful because it reduces the output of catecholamines. And the drug that I mentioned earlier, propranolol, might be helpful because it blocks the action of catecholamines after they've been released. And they're not antipsychotic. But I would only consider them if someone is really hyper. Not for anything else.
No, that makes sense. And mechanistically, that it makes sense how those work. And I like that there's at least mechanisms to back up the reasoning for using them.
True. And again, propranolol, it's contraindicated if someone has asthma. So just make sure that.
No, this has given so much information to anyone listening. I think they're going to take away a lot of information about, inflammation in general, mast cells, autism, how to help these kids. And
I also want to point out to everybody that the terms used to promote supplements can be very misleading. So, for instance, it's very popular to say immune booster. But if I have inflammation, I don't want to boost my immune system because inflammation is going to get worse. And that's an issue that I have, if I can say I have an issue.
Hopefully a lot of improvement to come. But I appreciate your input there.
Thank you. So collect some nice questions from the audience and let's have another session.
Absolutely. I hope we have a ton. This has been absolutely phenomenal. I am so glad we did this. Thank you again. I hope we can talk soon. And
God bless you and God bless your children and all the children.